Okay, it's a pleasure to be here. My first time in Israel, and I'm super excited to be here. But I also have to confess that I'm a bit of an imposter because I'm really not a neuroscientist, but rather my lab is interested in mechanisms of gene regulation in the context of development. And in particular, over the last few years, we got especially interested in human development and in human-specific aspects of developmental gene regulation. And mechanistically, we're interested in understanding how information is integrated on chromatin. In other words, how regulatory information that is encoded by the DNA, and specifically regulatory elements, is read in the context of the specific lineage, which can, by lineage, specific transcription factors that can sense their history, and also in the context of this specific cell signaling environment. And all major signaling pathways do in fact end on chromatin and with signaling effectors talking to regulatory elements. So we really want to understand how these three layers of information, our genetic information, context of a specific lineage, and then environment, work together to establish permissive chromatin states that in turn allow for very precise spatiotemporal control of gene expression during development. And when we talk about precise spatiotemporal control of gene expression, of course we have to talk about enhancers, and several talks today already mentioned enhancers. And these are these modular regulatory elements that are highly specialized for have highly specialized spatial temporal activity and can also act at long distances from their target genes. And they function as clusters of transcription factor binding sites, again representing both of these lineage-specific factors and the signaling effectors that together cooperate promoting gene activation. But over the years, we also learned that when active enhancers share certain common chromatin features, such as the presence of general coactivators like the P300 or mediator or remodeling complexes, and also presence of specific histone marks on flanking nucleosomes. And we also learned that these chromatin signatures, it's very simple combinatorial chromatin signatures, are very predictive of enhancer position and activity and can be used for enhancer annotation in a variety of cell types and species. The idea here is that we can use technologies such as ChIP-seq to map positions of germinal coactivators or this enhancer-associated histone modifications, and based on this information, we can infer the position and to some extent also activity level of enhancer in a given cell type. And indeed, these type of approaches have been utilized by many consortia now and also individual labs, and this revealed enormous complexity of enhancer landscapes. Current estimates are that our genome harbors over 1 million enhancers, spanning roughly 10% of the human genome. But a huge challenge for the field is to understand how enhancers encode human traits. In other words, how the genetic information that's contained within the regulatory elements reveals itself through developmental process to produce specific human phenotypes. And indeed, from many GWAS studies, we learned that many trait-associated loci do indeed map to the non-coding parts of the genome and are specifically enriched in developmental enhancers. But how do we make this genotype to phenotype connection, which essentially is a holy grail of developmental biology? So I would want to argue that the time is ripe to start making these connections due to progress that has been made in the field. First, our ability to systematically identify cell type specific enhancers. Second, through in vitro modeling, in vitro differentiation, organoids models that we already heard about today, we can finally model some aspects of human development in the dish. And while these in vitro models, of course, they have their own limitations, nonetheless, through follow-up studies, we and others have been able to convince ourselves that these models can nevertheless be very useful tools for understanding complex human phenotypes. And one of the phenotypes that we are really interested in is human face. And we've also been very inspired by the Evo-Devo work, showing that studying evolution of closely related species can be a very powerful tool for uncovering genotype to phenotype connections. So work from labs like Sean Carroll or David Kingsley unequivocally demonstrated that change in enhancer sequence can drive change in phenotypes such as wing patterning in Drosophila or skeletal changes in stickleback fish. And we wanted to extend this type of approach, Evo-Devo approach, to higher primates, and in what we termed cellular anthropology, use this in vitro stem cell differentiation models from humans and non-human primates to model regulatory landscape evolution with the with an ultimate goal of understanding the genotype to phenotype connection. And this is what I will tell you about today. Some of this work is published, but I will also at the end talk about some of our unpublished work on human facial variation. So I already mentioned that one of the phenotypes that we're really interested in is human face. And if we want to understand how human face forms in development, we have to go back very early when a very unique cell type called cranial neural crest cells is specified. So cranial neural crest cells is about as neural as it will get in my talk. But I hope to convince you, and I know that some in the audience are already convinced, that this is in fact the most interesting cell type. So, yeah. Of course, you forget the brain. So why is this interesting? These cells have several very interesting properties. So they are specified in the dorsal part of the neural tube, and then upon specification, they delaminate, undergo epithelial to mesenchymal transition, and they migrate long distances in the embryo. So they are sometimes called the wanderers of the embryo, so thought to be the most migratory cells that arises during development. But a very interesting feature of these cells is that they're also extremely developmentally or epigenetically, if you will, plastic. And they essentially expand their differentiation potential as they get specified. And in particular, in the cranial region, these cells, upon arriving at their destinations, can differentiate to over 100 different cell types, including bone, cartilage, and connective tissue. And this is very unusual because, of course, elsewhere in the body, bone cartilage is derived from the mesoderm. And here we have an unusual situation where there is ectodermally derived mesenchyme in the head region. And the contribution of these cells is such that, in fact, most of our face and head is derived from the cranial neural crest cells. And it's therefore not surprising that the cranial neural crest cells derivatives are thought to have played a key role in human evolution, facilitating not only uniquely human facial appearance, but also a number of adaptations associated with upright posture, speech articulation, and of course enlarged brain size, because the packaging for the brain has to co-evolve with the brain. But how to model this regulatory evolution and how to get access to these cells? So of course, in embryonic context, these cells are transient, arise about 3 to 6 weeks of human gestation. They're highly inaccessible. So we had to learn how to derive them in vitro, and over the last years, my lab and also Lawrence Studer's lab and a few others developed protocols where by mimicking natural developmental progression in vitro, we can derive cells that have molecular characteristics of the bona fide cranial neural crest cells, but also cellular characteristics. So for example, we can transplant these cells to the chick embryo, and they will migrate, assuming correct positional identities, and they also retain this very broad differentiation potential characteristic of the crest. And more recently, we extended this model to non-human primates, including the chimpanzee, and this was work done by Sarah Prescott, a very talented graduate student in the lab. And here a key for enabling our comparisons between species was actually minimizing heterogeneity of the population and properly staging them. And before we achieved that, actually most of the differences that we saw were developmental rather than interspecies. So this, I'm really not going to show all this optimization work, but I want to give shout out to Sarah for this heroic effort. But anyway, with this model in hand, she then used epigenomic strategy to annotate regulatory elements. So she's done ChIP-seq for transcription factors and general coactivators, ATAC-seq to map hypersensitivity, and also ChIP-seq for a number of histone modifications associated with active regulatory elements. And this allowed us to annotate active enhancers and promoters and distinguish these two classes of regulatory elements. So here is just a control showing that even though we're mapping these elements in an in vitro model, nevertheless, when put in the context of the mouse embryo in the reporter assay, they're able to drive craniofacial gene expression in vivo in the mouse, with different elements able to light up different parts of the face, suggesting that multiple facial identities actually arise in our model. But can we use then these epigenomic comparisons to experimentally map regulatory divergence in human and chimp neural crest? And the idea here is simple. We're comparing the same cell type between two closely related species, so most of the epigenomic patterns should be invariant. But yet we should be able to find some regions that are differential, either due to structural variation between the species at regions with no clear orthology, or due to quantitative change in activity at orthologous regions. So how do the actual data look like? Well, indeed, epigenomic patterns overall are very, very similar between human and chimp, but we're also able to identify the regions that bear either chimp-biased or human-biased chromatin enhancer signatures, as exemplified here. And we can quantify this across the whole genome and compare different individuals of the same species, and that's shown in red, or between species, between human and chimpanzee, and that variation is highlighted in blue. So this variation that falls outside of the intraspecies variation, I will from now on refer to our candidate human-biased or chimp-biased enhancer elements. So indeed, turns out that actually quantitative change at orthologous regions seems to be the prevalent mechanism driving epigenomic variation between human and chimp, with a smaller contribution, and probably we have even overestimated it here due to structural variation. So this source of variation begs an immediate question. Are these differences due to difference in the transregulatory environments of the human and chimpanzee neural crest, namely differences in transcription factor networks and other layers of regulations, or maybe we haven't even staged these cells properly? Or are these due to the cisregulatory changes, meaning differences in enhancers themselves? So to distinguish between these two possibilities, we used a derivative of the star-seq approach. So star-seq has been described by Allelk Stark's lab a few years back in Drosophila, and this is essentially a strategy which allows for testing of enhancer activity in the context of the reporter, and relies on the fact that enhancers can activate their own expression at a distance and then serve in the context of the reporter as their own barcode. So then we devised a library, synthetic enhancer library, that contained a number of elements that were either human-biased, chimp-biased, or invariant in our assay. And we included both human and chimp orthologous sequences, and then cloned this whole library to the vector and tested it in the context of either chimp or human cranial crest cells. And from these experiments, we found two interesting things. First of all, the bias in epigenomic signatures translated into the bias in ability of these sequences to drive gene expression in the context of the reporter. Or in other words, what we predicted as human bias was human biased generally in our assay, or chimp biased was chimp biased in the reporter assay. But another important conclusion was that the direction of the bias was the same, regardless whether we tested our library in the context of the chimp or human neural crest cells. And what this tells us is that the bias is encoded by enhancer sequence itself, rather than imposed by the differences in transregulatory environment of the human and chimp neural crest. So if it's in sequence, what is it about the sequence? And interestingly, even though we see these differences in activity, there are typically only few SNPs, several 3 to 5 base pair changes between human and chimp. So this gives us an idea that we can in fact use this interspecies genetic diversity like a large-scale enhancer mutagenesis screen that nature has already done for us. And the idea here is that since enhancers function as clusters of transcription factor binding sites, we can quantitatively measure which differences between human and chimp enhancer sequence give rise to differences in putative fit or putative binding affinity for these transcription factors, or in other words, p-value of the fit to their consensus motifs. And we can measure this quantitatively across different motifs and relate it to another variable, namely quantitative differences in epigenomic signatures such as K27 acetylation. And then we can correlate these two variables and derive correlation coefficient, and this is what's shown here in this heat map across different motifs and different epigenomic features. So what did we get from this type of approach? Well, we got what we kind of expected to get, which is a group of motifs that were weakly positively correlated with gain of active chromatin signatures, and not surprisingly, though, those generally correspond to known neural crest transcription factors. But we also got a group of motifs that were negatively correlated, namely gain of the motif negatively correlated or correlated with a decrease in active chromatin signatures. And interestingly, some of these motifs, at least the ones that we can unequivocally identify, are associated with known sequence-dependent repressors. So this suggests that the enhancer activity can be modulated in the course of evolution by plugging in and out regulatory motifs for repressors. But by far the biggest surprise we got from this approach was that there was a single motif that was an incredible outlier and was strongly predictive of changes in chromatin states, and could explain about 30 to 40% of all the epigenomic variation between human and chimp. And this corresponds to a long novel motif. We termed it a coordinator motif. And some other interesting feature of this motif is that it was also predictive of binding of other transcription factors with unrelated motifs. And moreover, it was enriched at neural crest selective enhancers as compared to those that were active pleiotropically. So we were scratching our heads, what it means. Again, in this type of analysis, you rarely expect to find a singular motif that would be most predictive of everything you look at. But then we noticed, it's my belief that a lot of the things in gene regulation have already been discovered in Drosophila, so whenever we're scratching our heads, I go back to Drosophila literature and see what's out there. And indeed, there's a precedent where a tag team, so-called tag team motif, has been described in Drosophila embryo that also has similar features. Mainly, it's highly predictive of binding of other transcription factors. And mechanistically, it turned out that tag team motif, which by the way looks nothing like the coordinator, so we think it's a conceptual parallel rather than direct parallel. So that the tag team motif binds the pioneer factor zelda, which then brings together a lot of multiple chromatin activating activities and essentially licenses enhancers for activation in early Drosophila development. So we really we think a compelling hypothesis here is that indeed a similar mechanism is operating in the context of the primate neural crest, and we're actively looking for the regulatory complex that binds coordinator. Recently, we convinced ourselves that it's actually not one factor, but probably a combination of two transcription factors, but we're still working on it. Okay, well, the discovery of coordinator also allowed us to really now look at coordinator changes as one potential important class of motifs whose change or whose evolutionary history we can reconstruct by looking at primate sequences and to reconstruct ancestral state, asking whether changes in coordinator motif represent chimp loss, human gain, chimp gain, or human loss during the evolution. And what we saw is that at the human biased enhancers, we saw prevalent contribution of either chimp losses or human gains, but very little human losses or chimp gains. And in contrast, at the chimp biased enhancers, which we saw the prevalent contribution of the human losses and chimp gains, but very little chimp losses or human gains. So this really validates our discovery, but also shows that the change in enhancer activity is equally likely to arise from the gain or loss during recent evolution. So it's really fairly symmetric regarding gains and losses. But now with Denisovan and Neanderthal genome, we can do the same exercise using those ancestral genomes, and not surprisingly, we discovered that most coordinator changes that happen between human and chimp happened prior to the split of humans from Neanderthals. After all, we're 10 times closer to Neanderthals than we are to chimps. But nevertheless, we did find a group of changes that appear to be either modern human or Neanderthal unique, and we're quite excited now trying to follow up on some of those enhancers to really figure out both their spatiotemporal activity and also whether these coordinator changes really indeed dictate the change in regulatory activity. And the reason why it's exciting is that we actually kind of know how Neanderthal cranial neural crest derivative looked like, because there are skulls and there are some morphometric measurements of the skulls. So idea is here we have a smaller genetic space to connect with smaller morphological difference space. So now, what about the spatiotemporal activity in the context of the embryo? So let me show you a couple of examples. Here is an example of a human biased enhancer that gained two coordinator motifs specifically in the human lineage. And now we're testing autologous human or chimp sequences in the context of the mouse embryo. Again, the trans circuitry is much more conserved than cis, therefore we can actually use a mouse embryo as a test tube or a readout for activity. And as you can see, where there's a shared expression in olfactory placode, there's also gain of activity in several domains in the head. And this is another example. This is more pan-neural crest enhancer. Again, epigenomically biased, strongly biased towards the human, and almost actually black and white difference in activity in the context of the embryo. So this is exciting because it shows we can systematically identify enhancers that gain or lost their regulatory activity during recent human evolution. But of course, it wouldn't matter unless we can link those changes to changes in gene expression. So we were very excited to see that genes near human biased enhancers in general tend to be human biased in expression, and those near chimp biased enhancers tend to be chimp biased in expression. And in fact, the more biased enhancers you have nearby, the more predictive it is of changes in gene expression, probably due to complexities and redundancies in enhancer landscapes. But these are not some random genes. In fact, when we do ontology analysis on these genes, we see that all top categories are craniofacials and actually associated with development of different craniofacial structures that are known to be divergent between human and chimps. And this includes genes with a known dosage-dependent effects on facial morphology. So let me give you an example of the chimp bias. We have here PAX3 or endothelin receptor A, both of which have been actually associated in a dosage-dependent manner with changes in facial morphology in humans. And one of my favorite examples on the human biased side is BMP4, because this gene has already been associated with variation in facial morphology, but in Darwin's finches, where increased BMP4 levels in developing cranial neural crest has been associated with changes in beak morphology. And moreover, elevated expression of BMP4 specifically in the neural crest cells result, when done in the mouse on the neural crest specific driver, results in jaw size reduction and rounding of the skull shape. And this is interestingly two features that are really quite characteristics of the human facial morphology. So this illustrates that perhaps some of the changes in regulatory landscape can be modeled in the mouse. So mouse is not completely hopeless model, and we're now following up on some of those by chimpanzizing or humanizing regulatory landscape. But we also have to admit that mouse is not a great model for studying human facial morphology and its variation. So what is the alternative? So the alternative is actually to harness normal range human facial variation. We all look different from each other. And it is a trait that we can actually precisely quantify, precisely and non-invasively quantify between different humans. And obviously in anthropology, that there has been a good tradition in morphometric measurements. It's also true that there are good arguments that facial form is largely genetically determined. Of course, monozygotic twins make a good case for that, but also we all know we look like our family whether we like it or not. So therefore, this ability to measure with the fact that it's highly genetic opens up for genome-wide association studies, and indeed our collaborators Peter Cleese, Seth Weinberg, and Mark Shriver have recently performed genome-wide association study of the population of European ancestry and identify about, 15 highly reproducible loci associated with normal range facial variation. And what is really interesting about their study is not just that they're identifying loci generally associated with variation, but they also divided those into very specific facial modules and measuring this facial variation specifically over unique parts of the face, and this is highlighted in those white patches. This is where different modules are associated with the most variation, and here are the 15 SNPs, and you can see different of those different SNPs are associated with different aspects of facial variation in humans. And not surprisingly, at least to me, that all identified SNPs are actually within non-coding parts of the region. So this leads us to sort of my favorite hypothesis, which is that allelic variants of neural crest enhancers may regulate variation of craniofacial features in humans, with the idea that we got some enhancer alleles from our mom and some from our dad, and it's the combination of this regulatory variance that gives us certain facial morphology that is clearly related to our family but also distinct. Okay, so when we take those GWAS regions and look at their chromatin marking patterns across 50 different cell types and including all our neural crest samples, we see that these GWAS identified loci actually preferentially marked by active chromatin marks in the neural crest. So this argues that indeed the variation arises early in development, already in these neural crest cells. And moreover, when we then cross-reference at higher resolution, this identified SNPs with different classes of regulatory regions that we see in the neural crest cells, we see enrichment for enhancers, including 3 of these regions mapping to the strongly active enhancers or in very close proximity, so they're within LD, but also a group of ones that are very close to enhancers that are poised or primed for activation in the crest, which again makes sense because one would expect that variation also arises once these cells start differentiating into bone and cartilage. And here is just one example. This is a SNP on chromosome 2 that is linked to variation in jaw morphology, specific aspect highlighted in yellow. And we actually, when we look underneath in our epigenomic data sets, this overlaps with an element that has active enhancer signature, K27 acetylation, P300, and K4 monomethylation. And to spice things up, this particular enhancer is actually strongly chimp biased in our data sets, arguing that perhaps an overlapping set of elements controls facial variation both within our own species, but also makes us different from other great ape species. So we're really quite excited about these observations, but these are still early days, and we're following up on them now. But overall, I hope I convinced you that with this approach of cellular anthropology, coupled to the genetic and morphometric analysis of human populations, hopefully we will be able to make this genotype to phenotype connection and do so directly in humans. And with this, I would like to end. So what I told you about, that we can now derive through in vitro differentiation models cell populations that are relevant both developmentally and evolutionary. Of course, this is not limited to neural crest, but I think it will be exciting to do similar type of studies in neurons and or perhaps in organoids if you can control for cell heterogeneity. And then we can use comparative epigenomics to systematically map regulatory divergence and identify candidate regulatory elements that change their activity. We can validate a subset of those using mouse embryo due to the higher conservation of the trans versus cis circuitry. And through the sequence analysis, I hope I can convince you that I've convinced you that we can really learn a lot about the cell type of interest, such as our discovery of the coordinator motif. Although, as I said, it's really going to be important to figure out what regulatory complex binds there. We can also learn about genomic features of species-biased enhancers. That's something I haven't discussed in detail today, but we're also learning interesting things there. And finally, we're hoping that this type of work can be a resource for morphological studies. And with this, I want to finish and acknowledge people involved. So almost all of the experimental work that I've talked about today was done by a single graduate student, Sarah Prescott, who now moved on to a postdoc at Harvard. And all the data analysis was done by Tomek Świegut. And I also want to acknowledge our collaborators, Rusty Gage for sharing chimp iPS cells, Vitra Larry for help with the mouse genetic work, and Peter Claes, Seth Weinberg, and Mark Shriver for the collaboration on the GWAS work. And thank you for listening.